For any assignment-related queries, call us at +1 678 648 4277. You can mail us at info@nursingassignmenthelp.com, info@nursingassignmenthelp.com, or reach us at https://www.nursingassignmenthelp.com. https://www.nursingassignmenthelp.com. Microbiology Assignment Help. Problem 1. A. Describe the different sorts of evidence for life's appearance on the early Earth. 3.5 Bia. Answer. The evidence for this phenomenon is isotopic record, rocks and microfossils, organic geochemical records, and molecular evolution. Microfossils exist that look as though they could be from cyanobacteria. Stromatolites have been formed from cyanobacteria and other algae-binding sediment together forming biolayers. Carbon isotope signatures from 3.4 billion-year-old mat-like structures suggest carbon dioxide fixation. The presence of stable carbon and oxygen isotopes also suggest bacteria were producing large quantities well before other oxygen-consuming organisms were present. Molecular signatures, such as steranes and 2-methylhopanes, only known to be produced by eukaryotes and cyanobacteria, respectively, have been isolated from extremely old rock. The presence of iron beds is another clue to the presence of bacteria changing the atmosphere over from mainly carbon dioxide to more nitrogen and oxygen. C. What dramatic change 2.5 billion years ago has influenced biology on Earth ever since, and how did it happen? Answer. Great oxidation event. Bacteria evolved that were capable of nursingassignmenthelp.com. Producing oxygen via photosynthesis, cyanobacteria. They pumped, and continue to pump, large amounts of oxygen into the atmosphere. Problem 2a. Describe how you might determine the stable isotope compositions of different compounds in inorganic, as well as organic or living materials. Answer. Isotope ratio description. Compare ratios of heavy and light isotopes of compounds. B. What elements might be useful for investigating biological processes using stable isotope ratios? Answer. Hydrogen, carbon, nitrogen, oxygen, and sulfur. C. Describe in detail the metric that's used to differentiate stable isotope content of a specific element in different compounds. Answer. Isotope ratios. R equals heavy isotope / light isotope. Mass spectrometry is used to compare the isotopic ration of a sample to a known standard. nursingassignmenthelp.com. Problem 3a. Draw the general structure of gram-negative and gram-positive bacterial cell walls. B. What gives these cell walls their structural integrity? How and why is that important? Answer. Structural integrity of bacterial cell walls is provided by peptidoglycan cross-linking, which is important for stability and flexibility of the cell to withstand stress/force. nursingassignmenthelp.com. C. What are the major differences and their biological implications? Answer. Gram-positive, no outer membrane, thus cell wall consists of only wall-anchored proteins, teichoic acids and lipoteichoic acid in the peptidoglycan layer. Gram-negative, outer membrane plus peptidoglycan layer, periplasm. The outer membrane consists of lipopolysaccharide, porins, lipoproteins, and phospholipids. Biological implications. Reasonable answers accepted. The outer membrane of gram-negative organisms allows space for many enzymes to reside outside the cell membrane, such as chemoreceptors and transporters, as well as porins on through the outer membrane. At the same time, endotoxins due to lipid of LPS are present in gram-negative organisms. Problem 4. A. Describe the various functions of the cell membrane in bacteria. Answer. The cytoplasmic membrane is important for anchoring proteins, acting as a permeability barrier, and for energy conservation. B. Name 3 different classes of transport systems. Answer: uniporters, symporters, and antiporters. NursingAssignmentHelp.com. C. Describe the 3 different functional types of transport systems and their general similarities and differences. Answer: simple transporters involve only a membrane-spanning component, i.e. lack permease transporter. Phosphotransferase-type transporters involve series of proteins that cooperate in the transport event, i.e. phosphotransferase system. ABC systems involve a membrane-spanning component, a periplasmic binding component, and AT-fibrillizing proteins, kinases. Problem 5. There exists a motile marine cyanobacterium that can swim at speeds of 80 but no one has ever identified its flagella, despite repeated attempts. Its mode of motility still remains a mystery, but its Reynolds R number is not. Assuming a swimming speed of 80 and a cell radius of 2000 nanometers, calculate R for this bacterium. How does this compare to E. coli? Answer: R equals A asterisk nu asterisk rho slash eta equals A asterisk nu slash upsilon, where is the length, upsilon is kinematic viscosity, rho is fluid density, nu is velocity, and eta is fluid viscosity. R equals 2000 nanometers asterisk 80,000 nanometers per second asterisk 1 slash 1012 nm2 slash SEC equals 1.6 x 10 to 4. R is comparable to E. coli. NursingAssignmentHelp.com. Problem 6. A. Draw and describe the structural and functional components and properties of bacterial flagella and their general mode of synthesis. Answer: flagella are long, spiral-shaped rods anchored in the cytoplasmic membrane and are attached to the cell surface by a complex of flagellar motor proteins, which include L and P rings anchoring the hook to the outer membrane and peptidoglycan, moat proteins, MS and C rings that surround the FLI proteins, motor switch. Located in the cytoplasmic membrane, there is a hook in the flagellum filament that adjoins the flagellum and flagellar motor. Flagellum is a multimer of the protein flagellin. Synthesis begins with the motor proteins, then the hook ending with flagellin monomers to grow the flagellum. B. If you were to follow the synthesis of flagella with a pulse chase experiment, by adding first unlabeled amino acids followed by radio-labeled amino acids, what pattern of labeling during flagellar biosynthesis would you expect to see? Answer: A, B, or C, A. Flagellin monomers added to tip, not base. NursingAssignmentHelp.com.